Good afternoon, and thank you for joining us. I'm Dr. Gail Boland, Director of the STD Prevention Program at CDC. The Division of STD Prevention and the American Sexually Transmitted Diseases Association are partnering to bring you the latest research and program innovations for STD prevention in the United States with the STD Prevention Science Series. I'm very excited about this partnership and the opportunity to share the cutting-edge work colleagues are doing. Through this quarterly series, we will be presenting emerging research topics and best program practices for those interested in STD prevention, including HIV. I hope that those working in state and local public health programs, those at academic institutions, and others in STD prevention field will join us. Our goal is to bring science and program excellence to you in a timely fashion via the STD Prevention Science Series. I will now turn it over to the coordinator of this series, Dr. Sevgi Aral, the Associate Director for Science in the Division of STD Prevention, to moderate today's session. It's not just the pathogen anymore, the genital microbiome and implications for sexually transmitted infections, presented by Dr. Jeanne Marrazzo.
Good afternoon. It gives me tremendous pleasure to introduce Jeanne Marrazzo as the first speaker in the series, which I hope is going to be incredibly exciting every single time. Jeanne is Professor of Medicine in Infectious Diseases at the University of Washington in Seattle. She's the Medical Director of the Seattle STD HIV Prevention Training Center. She received her undergraduate training at Harvard-Radcliffe, her medical degree at Jefferson Medical College in Philadelphia, and her MPH at the University of Washington in Seattle. She did her internship and her residency at Yale New Haven Hospital, her fellowship at the University of Washington. She's board certified in internal medicine and infectious diseases. She is a member of the editorial boards of Sexually Transmitted Diseases, Sexually Transmitted Infections, Journal of Infectious Diseases in Obstetrics and Gynecology. She is an associate editor of Sexually Transmitted Diseases. She is the immediate past president of the American STD Association. She has numerous honors, which I'm not going to go through because I don't want to take away her time, and numerous publications, which were too many to count. She likes sailing. She also likes good wine. Here is Jeanne Marrazzo.
Sevgi, thank you. I knew I could count on you to give a true insight into my character. So I want to thank you for that, and I want to thank Gail in absentia for that lovely introduction. It's really exciting to start off this series. I'm extremely excited to be here and very happy to have this turnout, both here and online. I don't know if it went away, but up there were 126 people listening in online, which is pretty cool for our first effort. And I hope you've had a chance to take a look at my images while Sevgi was talking. I love this photo on the left, which a friend of mine who knows my obsession about all things vaginal took for me not too long ago. She found these suppositories in some antique store. And it was a product that was made in 1919, and they were called Madame Dean's Vaginal Suppositories, contain ingredients of unquestionable merit. And in 1919, a box cost 50 cents. And I think, to me, that speaks to a measure of how desperate women probably were, even then, to get their vaginal complaints addressed. And I guess the question I would pose is, are we really in a different era today when you look at 2012? And I'll tell you what that picture is going to be in a little bit. And I would say we are better off in many ways, but when it really comes to the reality of women's vaginal health, I'm not quite sure that we have made immense strides. So let's go ahead and sort of talk about what that is. People usually save the acknowledgments for the end of these talks, but I really could not be doing this talk without the incredible laboratory collaborators who've done most of the work that I'm going to present. David Fredericks, in particular, has been the genius behind the molecular analyses that has informed our ability to do clinical and epidemiologic assessments of the vaginal microbiology. And his staff, as well as the incredible clinical staff who run my projects at the University of Washington, are shown there. So really have to acknowledge them right up. So Steffi, I don't think you could have timed this lecture any better. If anybody who saw the paper last week, listened to NPR, saw the news, you would have heard that there were several publications that came out on the Human Microbiome Project. And one of the most striking, I think, reductive, simplified statistics that really grabs people's attention is exemplified in the diagram in that upper right-hand corner. And it really points out that when you look at the genetic material in the human body, the human genome proper represents about 23,000 genes. And when you look at the human microbiome, meaning the microbiome that is existence in bacteria in the human body, there are more than 1 million genes. So the vast majority of genetic material in your body and in my body really belongs to the commensals, the bacteria that lives there. And I think for people who haven't been following this field, sort of your average newspaper reader, your average NPR listener, that was a pretty surprising fact. So keep that in mind. So there's been quite an evolution in the whole field of the human vaginal microbiome. What I want to do in this talk is to look at BV as a framework for this discussion, bacterial vaginosis, and I'm going to define that for those of you who aren't familiar with it. I'll talk about what the consequences are of that condition. Why should we care? Does it really matter? And then I'll talk about the implications of the microbiome approach, specifically for BV, with relationship to the pathogenesis of the national history and the role of sexual behaviors. We are in the STD division, so I think it's important to think about how it relates to our work as we look at STD-related syndromes and pathogenesis of STD infections. And then, I'll again talk a little bit more about the implications for diagnosis and, as I mentioned, some specific syndromes. So this is what people were talking about last week, and that image on the right side, Tending the Body's Microbial Garden, was in yesterday's Science Times from the New York Times. And I love the quote by Carl Zimmer: as we learn more about the trillions of microbes we carry around, some scientists want to be like wildlife managers, nurturing the good and weeding out the bad. And this is really getting away from the idea of germ warfare that we have waged since the dawn of antibiotics, right? It's really been about controlling the bad things that invade our bodies. And I think shifting that paradigm is really what this is about. And when we think of pathogens and STD pathogens, I don't think we can separate them out anymore from what's going on in our bodies and what we're exposed to when we have sex with other people. And that's really where I'm going to go with that. 3 of the most important papers are listed there. Those are the papers that were in Nature, although there were a number of papers in the PLOS journals as well. And I think if you are new to this field, taking a look at David Relman's editorial called Learning About Who We Are is probably a great introduction to what this really is about. The other 2 papers are fairly technical, but really for those of you who want to delve into the gory details, feel free to take a look. So what is an optimal vaginal milieu, and what is bacterial vaginosis, which is what I think of as the opposite? Sorry that the spacing there of those boxes is a little bit weird, but what you're looking at on the left-hand side, and let's see, oh good, it actually works, is a gram stain of normal vaginal fluid. And what you can see here are all these lovely, long, lanky lactobacilli. Those are very, very purple. They're gram-positive. And that's what a very healthy lactobacillus predominant vaginal environment looks like. It's associated very strongly with a low pH, quite low, less than 4.7. That's compared to your blood pH, which is about 7.3 to 7.5. So very, very buffered. And that happens because of the lactic acid produced by these lactobacilli. And this is sort of a prototypical Nugent score of 0. We use a Nugent scale to measure what the lactobacillus, i.e. healthy predominance, is relative to this situation, which is frank BV, in which you've really lost all those lactobacilli. As you can see, the morphotypes that represent them are now gone, and instead you've got a lot of pink and purple dots, some gram-negative, positive rods, but nothing that looks like lactose. And that's really the anaerobic predominance that characterizes BV. We can also use the AMPA criteria, which are clinical criteria, to define BV, and they're defined by abnormal pH, abnormal discharge, abnormal odor, and then clue cells, which I'll show you in a second, on vaginal microscopy. So for a long time, we've known that BV represents a profound loss of hydrogen peroxide producing lactose, primarily L. triphlatus and L. ginsenii, and an overgrowth of these commensal anaerobes, but really that's all we knew and that's all we thought about. But in the intervening, probably last 10 years, we started to define this as a very immune dysregulated state. There's a lot of disruption of normal cytokines, which are listed there, which I won't go into, but you also lose some things that are very important for defenses against superinfection with STD-related pathogens. So, for example, SLITI, or secretory leukocyte protease inhibitor, very strongly protective against HIV, has a lot of virucidal activity, and really is a very important innate defensive, really almost disappears when you have BV. So the clinical and epidemiologic feature is probably familiar to many clinicians who are listening in. On the upper left is the classic discharge, and on the upper right is just a reminder that the amine odor is a byproduct of these amines being metabolized. And that fishy smell that people smell when they have BV really represents just the metabolism of these anaerobes and the volatile amines that they actually produce. It is the most common cause of vulvovaginal complaints in the world. It's very prevalent particularly in areas of the world with very high HIV incidence. In some of the areas where the HIV prevention studies are being done, prevalence of BV approaches 75 to 80%. And in many of those places, the incidence of HIV, as we'll talk about in a moment, is really quite staggering. Epidemiologically, very consistent and strong associations with race, very, very high in African American women. Unprotected sex with male partner consistently predicts BV, as does sex with a female partner. And we'll come back to some of those as we talk about the microbiome. Abnormal discharge and odor, often present, but sometimes not. And interestingly, when you treat these anaerobes with an antibiotic, they generally respond, they usually go away, but a fairly substantial minority of women will fail early treatment, 15 to 20%, and even if they respond initially, almost everybody recurs in the year following diagnosis. So very, very difficult to recolonize with a healthy lactobacillus predominant flora, and very big issue with the microbiome. Well, BV is not nice to have. It doesn't smell good. It doesn't feel good. But it really got a lot of attention because it became increasingly and consistently linked to adverse outcomes related to reproductive health in the 80s, early 90s. And this slide just points out that there are a number of pregnancy-related outcomes, as shown there, that have been pretty consistently linked with BV. In fact, some estimates early in the 90s thought that about 11% of preterm deliveries may be attributable to BV. I don't think that estimate has been updated, so I'm not quite sure if it's still accurate. But I think people believe that it still is substantial. Infection with other STDs, some very nice prospective work, particularly by the Baltimore group, has shown that having abnormal vaginal flora is an independent predictor of subsequent likelihood of establishment of gonorrhea, chlamydia, tricks, HSV, and HPV. And then I want to talk a little bit about HIV transmission because I want to engage those of you who are skeptical about BV and its importance and who really maybe only care or preferentially care more about HIV early on. So let's talk about that. What happened? Very nice meta-analysis by Julius Adishili looking at BV and HIV acquisition, published a couple of years ago, and looked at a number of pretty good studies, a number by Taha and a couple of other folks, largely in Africa, looking at the relative risk of HIV acquisition after a BV diagnosis. Now, it's a complicated thing, as we'll hear about later, because BV comes and goes. It's a dynamic situation, so you have to view, I think, these studies somewhat, carefully. Nonetheless, the weight of the evidence suggests that there is a consistent increase in risk in these analyses with an overall estimate of 1.61 for the relative risk. It may not sound like much, but when you look at the prevalence of BV, as I mentioned, being about 70%, and the incidence of HIV being very high, the population attributable risk for BV has been estimated around 20 or 25% when you're looking at HIV acquisition in some of these countries. Some of the thoughts about why that might be are shown there. It could be that you don't have hydrogen peroxide because those lactobacilli aren't there to produce it, and that's one of the things that's going on. It could be that the alkaline pH activates local CD4 cells to be more receptive to getting infected with HIV, cytokine upregulation, or the anaerobes that predominate in BV may actually stimulate T cells and monocytes, encouraging them to express HIV or be, again, more receptive to HIV. However, even if you're skeptical about this data, I think you will find the next set of data, which have not been published yet, but were presented by Craig Cohen in Rome last summer, really very interesting and intriguing. BV has also been independently associated with an increased risk of HIV transmission to uninfected male partners. It's not published yet, but I think it's very interesting. Where is this data from? Many of you probably have heard about Connie Kellum and Jared Baton's Partners in Prevention trial. It was the trial framework that looked at suppressive herpes therapy to prevent both HIV acquisition and transmission, and more recently looked at the delivery of oral pre-exposure prophylaxis in discordant couples and successfully showed that, or showed that that was very successful in reducing transmission in those couples. They had the opportunity to look at HIV acquisition in male partners who were the seronegative partner of seropositive couples by the woman's BV status over the course of the trial. So they could actually look longitudinally at sort of how often she had BV, how often she had normal flora, and then look at that, controlling for a number of other factors to see if it contributed to the male partner's HIV acquisition. You can imagine this is a very complicated analysis. There's a time-dependent variable. There are some fixed covariates that they used there. But I would say that this is fairly rigorously done. And what they showed is shown here, that if a woman had BV at the pre-visit where the man was ultimately tested for HIV, the hazard ratio for HIV incidence or detection was 3.6. That was even higher if the woman had BV at the current visit, and it was very high if she had a very high Nugent score. In fact, the hazard ratio there was 7.19. So there seems to be some gradient with abnormal flora. In other words, the risk went up for HIV transmission as the woman's time in a very disrupted vaginal environment defined by the bacteria was sustained. So fascinating. How could that be? What is that about? How could this possibly be affected? Some interesting hypotheses that this group has raised. Well, maybe BV actually increases the female genital tract concentration of HIV, making the women more directly infectious. They measured that. They showed that the women who had BV had a modest increase in genital HIV. It's probably not enough to really explain the marked increase in some of those hazard ratios that I showed you. Could BV increase the proportion of infectious HIV? We know the lactobacilli and their products directly kill HIV. Maybe, we don't know. Or could BV indirectly increase HIV susceptibility in a male partner? And because this is a microbiome-focused talk, I want to just point out that people have shown, using some of these cultivation-independent methods I'm going to talk about, that female and male genital microbiota are shared. So could the BV bug actually be stimulating Langerhans cells and CD4 cells in the male, whether they're a circumcised male or an uncircumcised male? Could that be partly making the environment more receptive? Could the BV bug be establishing colonization/infection? I don't know that that's what we're talking about in these men. These are all areas that are under investigation, but very robust, biologically plausible findings with a gradient by abnormality of vaginal flora. So let's take a step back now that I've hopefully piqued your interest about some intriguing, perhaps, parts of BV, and talk about the microbiology. What is the microbiome? What are we doing? We've known for a long time, as I mentioned, that using traditional cultivation approaches that a spectrum of anaerobes, as I said, characterize BV. I won't go through this list in the interest of time. I will just point out that everybody's heard probably about Gardnerella vaginalis. In fact, BV in the old days used to be called Gardnerella vaginitis because people thought it was an infection with Gardnerella. And if you biopsy the epithelium of the vagina in women with BV and you stain specifically for Gardnerella, as is done in that upper picture, you can find an incredibly thick adherent biofilm that's packed with Gardnerella in many women with BV. So it's heavily represented in biofilms associated with BV, and it's also universal. You find it all the time in women with BV. The problem is with Gardnerella is that you also find it in women who don't have BV. It's a colonizer present in about 35 to 50%. Same thing with some of the other bacteria that are discussed there. So I think we've known that some bacteria are characteristic, but there was never an ability to say that a single bacterium or consortium of bacteria appeared to explain BV as an infection. So the question of BV as an STD became very difficult. So could you use better methods, more sensitive methods, cultivation-independent methods, to detect BV-associated bacteria, recognizing that anaerobes don't like air, and it's very difficult to cultivate some of these extremophiles. So how do you do that? Thinking about describing the vaginal microbiome really involves that very approach. And if I had to take a step back as a non-molecular person and just sort of outline what we think has happened over the past few years, this has been the process. First of all, people have used broad-range 16S, bacterial 16S, ribosomal DNA PCR. I'll show you what that is in a minute. Then they have used visualization and confirmation with something called FISH, fluorescence in situ hybridization, which is that beautiful picture there, and was that picture on the first slide, and I'll tell you what that is. Once you've done that, you can design specific PCR assays to go after the bacteria that were defined or at least highlighted in that early broad-range survey, and then you can design quantitative PCRs to figure out exactly how many of them are there and when they're there. That's very exciting, and it got us very far, but we're now going even further with more intensive abilities to more quickly characterize the diversity, and pyrosequencing is sort of the next step, which I will mention very briefly. So don't freak out if you're not a molecular person. I'm not either. This is sort of my very basic description of 16S ribosomal RNA, but people should at least know what it is. It's basically a gene that's present in all bacteria. It codes for a small subunit of ribosomal RNA that you have to have for protein synthesis. You can't be a bacteria and not have this gene. The gene is very useful, though, because it has conserved regions, shown here in black, that are the same across all bacteria. So you can basically use those regions as little magnets to fish out bacteria. You won't know who they are specifically, but you'll get a bunch of them. You can then go in and use these variable sequences, which are shown in white there, to specifically figure out who is there individually. So what individual bacteria species are there. And that was really the first attempt to clarify what was going on in the vagina. And these next two slides are about the result of about 4 years of work on my colleague David's part. These were women that we enrolled in studies back in the late 90s and early 2000s, and this was our New England Journal paper that described the first attempt to really define, using the broad-range approach, who was present in women with and without BV. We hypothesized that using these approaches that we would find a lot of these BV bugs in women who didn't have BV. That didn't turn out to be the case. These are a number of women, 13 women who did not have BV, and these are the results basically of the analysis looking at that broad range. And what you can see, and I won't go into what the number of clones means, you can just sort of take it as a representation of the community members in these women. But you can see on the left-hand side that the vast majority of bacteria were represented by two Lactobacillus species, Lactobacillus crispatus, we weren't surprised about that, and Lactobacillus inners, which we were somewhat surprised about. We knew that inners was around. We didn't know what it does. We still don't know what it does, but it's around a lot. Nonetheless, we found only 16 species, which was a little bit paltry, we thought, and there were only 3 species per subject. So a remarkably homogeneous, kind of uninteresting, I would say, environment in women who did not have BV. So I think it confirmed what we already knew, although we had to ask, what is this thing called L. inners? I'll come back to that. And where are the BV-associated bacteria? Where do they come from? Do they just materialize out of thin air? Are they being delivered sexually? Do they come from the rectum or other anatomic features? Who knows? What about when we looked at women with BV? This is what we found, a very different picture. Hard to fit all the bacteria on the slide here. There were 20 women with BV, and David and his colleagues' team identified 46 species with an average of 14 species per subject. So a very rich environment. You can see the Gardnerella on the left-hand side very well represented, and then a number of other old friends there that you may have heard about, including Adipobium, Leptotrichia megaspira, etc. But I think one of the more striking things was that novel phylotypes, meaning things that had not really been described before, made up 58% of the clones in this library. So the diversity was greater than we expected, but the novel component was considerably more great than we expected. There were also a couple of things that you can see down the bottom there. I should have highlighted them, BV81, BV82, and BV83. These were bugs that were common in these women, but had never been speciated or sequenced before. So we were very interested in those bacteria, and David, in fact, developed those fluorescent in situ micrographs that I mentioned, and what they do, or what they represent, is the attachment of these labeled probes targeting specific bacterial ribosomal RNA. So in this slide, you're taking a smear of vaginal fluid. David made probes specifically to this thing called BV81, which is shown in green there, and you can see on that curve, on that little inset, it looks like a curved rod. And anybody know what that might remind you of? Anybody ever looked at mutant spores, the gram stains in the room? Looks a lot like Mobilungus, actually, which has been described as a player in BV for a long time. It turned out not to be Mobilungus, but to probably be confused with it on gram stain, interestingly enough. And then there were a couple of other things, BV82 in red there, and you'll see a few more. This is basically a high-tech clue cell. So it's a vaginal epithelial cell. Clue cell is what you see on a scaling microscopy. It's anaerobes, basically bacteria coating the clue cell. And you can see here that using these probes, you basically show the epithelial cell completely coated by BVAB1 and BVAB2. Interestingly enough, these bacteria are most closely related to bugs that live in the GI tract, particularly in rumens, so cows and bovine animals. And if you look sort of when this was starting out, most of this work had actually been done in animal husbandry and veterinary medicine, because clearly keeping the GI tract healthy in cows is economically a lot more important. Well, I won't go there, but you know where I'm going. And then BVAB2, we could find absolutely no close family affiliates. And then BVAB3 we just liked very much because of this really cool shape. It looked like a little wasp waist. And first, when we saw this, we thought that it actually might have been sporulating because it was a very close relative of the Clostridiales, or Clostridia bugs, which you may know from Clostridia perfringens, Clostridia sordelii, and a bunch of other things. And I'm going to show you some really interesting data to say that this bacteria, when present in BV, is strongly associated with cervicitis and cervical inflammation. So we're getting into this thing where does a dysbiosis that is represented by something like BV, could that actually represent something that's abnormal enough, disruptive enough, to start looking like some of the syndromes that overlap with our traditional STD pathways? And that's really, I think, a fascinating area for further discussion. Now, I'm not going to take you through the pyrosequencing approach, but trust me, what happens in this very exciting and interesting area is that you start out with that broad range approach that I mentioned. You use the 16S ribosomal RNA gene to, again, suck out those bacteria like a little magnet. You then put fusion primers on there, and you end up doing this specific PCR on this bead that allows for individual deposition of the beads in this plate. And then they are synthesized. It's really quite an amazing, I'm sure David would be mortified at my reductive description of that, but it's an amazing tool that allows you to get incredible amplification at incredibly specific levels, and it gives you just an unbelievable array of data. In fact, it gives you so much data that the major challenge is the informatics component of it. The reason that this particular paper, which should be coming out very soon, took so long, is that we had to work, David's team had to work very closely with the bioinformatics and molecular bacteria people at the University of Washington in the Hutch to try to figure out what the heck to do with all this data and how to classify it. So they basically had to develop a classification pipeline that would take advantage of all this molecular sequencing data. What do you find when you apply this even more sensitive and crazy descriptive method to these samples? You find even more. You find even more bacteria. You find even more unusual bacteria. And again, it's sort of just another example of the more you look, the more you find, and it's not really clear where it's going to end. The other thing that it allows you to do is some very, very novel assessments of the communities by various parameters. So this is just one example of what you can do. In this slide, each of the horizontal lines here, so each of this, for example, is an individual subject. And when you look over here in this vertical column, you're looking at the Ansel criteria score. In red, it means that this woman had BV by Ansel criteria, and means BV was present by Nugent criteria if it's red, and green is neither was present depending on the column there. And what you can see here is that these women really have, as we would expect with BV, tons of diversity. And these are all the bacteria that are included here. Quite a bit of them are other. And women without BV have these clusters that are really dominated by Lactobacillus and Lactobacillus crispatus. Not surprising. You see a lot of Lactobacillus inners. In particular, you see Lactobacillus inners in these people who are intermediate between BV and abnormal flora, suggesting that what may happen in this critical time is that there may be a vacuum that you lose the good lactobacilli, Lactobacillus inners moves in or moves out, and then it's really taken over by all these crazy anaerobes. The other thing I would say is that even within BV, though, you start to see specific little clusters. So these women clearly are different than this woman, these women down here. This one is dominated by Senecio leptotrichia species. These are dominated by Gardnerella. So you get the sense that not all BV is the same. And I think when you take a step back and realize that not all BV is symptomatic, not all BV causes abnormal outcomes of pregnancy, not all BV causes many things, you have to wonder, could this partly be defined by the specific microbiome? Clearly the host has got to be a player, but I think this kind of analysis ultimately is going to be a fingerprint for us to look at sequelae going forward. So you could imagine a study that would look at adverse outcomes of pregnancy that would prospectively look at the impact of careful characterization of the microbiome on whether or not adverse outcomes of pregnancy occur. This can get even more wild, and I will spare you the details, but I just want you to show what you can do. This is a heat map that shows correlations of these bacteria that David and his team generated. And you can see that the same bacteria are pretty much represented right next to each other, and what you're looking at here is whether they occur together. And not surprisingly, the lactobacilli are correlated with each other, as are the BV-associated bacteria. You would expect that. But among the BV-associated bacteria, there are also subgroups with strong positive correlations. And this again speaks, I think, to this argument that there may be communities that occur together and maybe are synergistic in affecting some of these adverse outcomes. Now, we're not the only folks who have sort of delved into this and tried to classify. Jacques Ravel's group at the University of Maryland has published some work really looking at vaginal community state types, dominated, as I note here, by a number of specific bacteria, and generated very similar kinds of visual tools to tell you what's represented by various measures. So, for example, in the C panel here, they've actually categorized things by Nugent score. So this is sort of the BV axis, if you want to call it that, and this is the normal Nugent score, so the women down here are more likely to have BV. So everybody, I think, is using informatics in a very novel way to look at incredibly complicated populations. Do these results have anything meaningful for us right now as diagnosticians and clinicians? Well, certainly many companies think they do. They're very excited about results like this. Remember, I told you that Gardnerella vaginalis, very nonspecific for BV, present in almost all women with BV, but also present in about 35% of women without. In contrast, if you look at bacteria like BV81, 2, or 3, as I mentioned, or Megaspira or BV82, you can use those assays to get incredibly specific predictors of BV. So take a look at the Megaspira or BV82. You can see that if those were assessed in BV-negative women, they were present only about 8% of the time, but they were present in 99% of women with BV. So very, very interesting. You can do the same thing with some of these others. So I can tell you that people are very interested in trying to move into molecular diagnostics for BV. The question is, what is this going to tell you in terms of treatment? Should we go there? Does this really mean that you're targeting the bacteria that are important for the adverse outcomes, whether it's symptoms or sequelae? And I think we just really don't know. Now, what about the natural history and pathogenesis? The next 3 slides, I don't want you to freak out. They're very busy. I will walk you through them. But this is a complicated topic, and there's no way to sugarcoat it. You just sort of have to look at this over time. Because one thing I haven't introduced in the discussion yet is the temporal axis, and that's where it really gets crazy. And I also want to just remind you of this plastic triangle, which those of you who are infectious disease oriented will remember. When I teach medical students and residents often, and we're trying to figure out what's going on with the patient, I often put this up. And I say, well, let's talk about the host. Where do they come from, and what pathogens can cause a compatible clinical syndrome? Can we use this framework for BV? I don't know, not so much. Again, this is a traditional pathogen-focused model for disease, and if we're really thinking about shifting our mindset to more of a dysbiosis and a kind of community situation, we may need to rethink this. Okay, so this is the scary slide. So let me just tell you what you're looking at here. These are data that Sujatha Srinivasan in David's lab published last year, I think, or maybe the year before. And what David and Sujatha and our group did was to follow women. We asked them to self-collect daily vaginal swabs for a period, in the first case, of 2 weeks, and then weekly thereafter. And that's, the axis is actually pretty similar for both of these women. And we validated this as a very good measure for performance of the quantitative PCR assays. And now I'm moving you into that third step, beyond FISH, beyond broad range, to quantitative PCR assessment. So now you're getting a sense of how many people, or how many, sorry, I split into anthropomorphization because I like the bacteria so much, how many bacteria were actually present in the vagina at a given time. So these women, very importantly, never had BV during the entire time. And we know that because we had gram stains on them for every day. And you can see their Nugent score was 0 throughout. So here's participant A. In the purple here are her levels of Lactobacillus crispatus, the good bacteria. And you can see she had over 10 million bacteria throughout the course of her month, or is actually a month. What is this? This is basically just a measure of 18S RNA, so it just makes you confident that you actually got cellular material. What I want you to know, she looks quite perfect, actually, throughout this, very consistent, exactly the kind of participant you would like to have in your studies, because you don't want anything messy when you're trying to figure this stuff out. But here I just want to show you that she has her menses in the last week. She never developed BV, but she has this spike. And what is that spike? That's Gardnerella vaginalis. Her lactobacilli actually come down. They never come down below 1 million copies. This never gets up to something that we think is probably significant, and she recovers very nicely. So this gives you a sense that something could be going on here. Now, this woman, on the other hand, is completely chaotic, despite the fact that she never developed BV during this whole time. So you can see she's got Lactobacillus crispus, again in purple, but her Gardnerella vaginalis concentrations, again, in red here is menses, and we had daily swabs during her menses here. You can see as soon as she starts to menstruate, Gardnerella plumps up here, and then when she stops, comes back down, and the Lactobacilli attains predominance. Again, she really has a very significant reversal of these two bugs, but she never developed BV. Strong association with menses in both cases, which I think is quite interesting. And then a number of other bugs that are shown here, too. Again, you can really look at the menses effect using these studies quite nicely, and we've known there's an association with menses as well and STDs for a long time. Gonorrhea is a classic pathogen that is thought to be, again, the thinking is always been that if you look at it in the lab, there's an iron affiliation, and so blood is iron, blood agar, et cetera, et cetera. But is that really all that's going on here? Could there be some other factors that relate to fluctuation of the vaginal bacteria around menses? Again, I won't take you through these slides because it's really the same thing, but it's dissecting that menstrual relationship a little bit more closely. We've also looked, of course, at reported unprotected sex during these daily diary sort of things, and we actually do see some interesting associations, but they're not quite ready for prime time. Now, what about women with BV? This is what happens, and I want to point out that this is also not predictable, and it speaks to our, I think, haphazard attempt to treat these dysbioses. We use anaerobic therapy, but, we're sort of just throwing something very nonspecific at these women. This is a woman who came in with a Nugent score of 8. Actually, they both had Nugent scores of 8. This woman was cured. This woman was not. So she gets her metronidazole here, shown in green. Again, she is having her menses here, not surprisingly. She gets her antibiotic here. Her Lactobacillus jensenii actually comes up very nicely, and she reestablishes Lactobacillus predominance very well, although she's still got a lot of Gardnerella even when things are finishing up here. This woman, on the other hand, got her antibiotic. She had attempts to sort of reestablish normal flora, but never managed to do it. So what these kinds of observations can really help you do is to dissect out what is accounting for failure to reestablish a healthy vaginal environment, at least from the microbiologic perspective. So can it help us figure out how women get or maintain BV? Because, ultimately our ultimate goal is to really create an environment that is protective against getting super infected with really bad things like HIV, gonorrhea, chlamydia, trick as well, and to have healthy babies as an opportunity, and to have healthy sex and not transmitting. So why do women get or maintain BV, and can this help us a little bit? Clearly, there could be a host factor. There's a lot of intriguing, I would say, early data to say that some women may not be able to block induction of these inflammatory factors that these bugs may stimulate. That may be determined by TLR, or toll-like receptor status, which is an endogenous innate immunity thing. Hormones. What about hormones? Huge relationship between hormones and STD, HIV acquisition, all kinds of interesting stuff. Clearly, something's going on in the perimenstrual period that also could relate to hormonal status. It may not just be the blood that's actually nourishing the bacteria or changing. Maybe it's the premenstrual shift in local hormones. It's actually stimulating some of these effects. We don't know. Menses independent of hormones. And then the biofilm hypothesis. The biofilm research is the other major frontier that I think as STD people we need to sort of start thinking about, because biofilms clearly are impenetrable for antibiotics, particularly oral antibiotics, and are a great focus of persistent infections, for catheter-related infections, but probably also for BV. And then the questions I want to get to for this group really are the role of sex. And can you use the microbiome do analyses to further help us, because a lot of the questions I get in the STD clinic, especially from women, are, but is this an STD? Did I get this from my partner? What should I tell my partner? And, we really don't know. We know that unprotected sexual activity is well described as a risk for BV in prospective studies. Condom use has been associated with reduced recurrence. In a study in Peru, James Webster showed very early that if you use condoms more frequently, or your partner does, you're more likely to sustain normal flora over time. And then there's the circumcision data, which I think is fascinating. So if you look at the Rakai trial of circumcision, that's one of the 3 trials that showed circumcision prevented HIV acquisition in men, they studied women whose partners were circumcised in that study, and they followed them over a year, and they related their incidence of BV at 1 year, their incidence of trick, to whether the men had been randomized to the circumcision arm or to the deferred circumcision arm. What do they find? The incidence of BV was actually reduced remarkably, about 40%. Wide confidence intervals, but pretty robust, I think, with this group in particular. Trichomoniasis as well, although not quite as significantly, but it was significant. So what happened? Actually, they studied the anaerobic microbiome of the penis in these men pre- and post-circumcision. And what they showed was that post-circumcision, again, it's common sense, but nobody had ever really shown it before, was that there was considerably less anaerobic diversity, and that makes sense. If you don't have the foreskin, you don't have the subepithelial area, which is probably relatively hypoxic compared to the surface skin. It's a very different mucosal environment, and so probably that was an excellent place for these bacteria to be harbored. Could that have been a reason that women weren't getting, quote-unquote, reinfected with these BV pathogens? I think it's a great hypothesis. However, remember that sex isn't always simple. People have different kinds of sex. They have different mucosal environments. And in fact, we know that anal sex is a consistent risk factor for BV. Not everybody asks about anal sex, of course, particularly prior to about 5 years ago. But almost everybody who's asked about the anal behaviors in studies that have looked at BV have shown a relationship. Is that surprising to you? Well, when you think that the anorectal environment is a much more sort of anaerobic environment, and you think of the gut, perhaps it's not. We also showed, as did Cass Feathers and Kat Bradshaw's group in Melbourne, that oral sex and a new partner with a history of BV in women with sex with lemon were the only significant risk factors for acquisition of BV. And we also showed that women share unique lactobacilli strains through sex. We used rep PCR fingerprinting in Sharon Hillier's lab to show that women clearly share lactobacillus identical isolates, and they're more likely to share depending on how long their relationship was. So the length of relationship really supported that they were sharing these bacteria. So I think in those women, for sure, the transfer of vaginal fluid with high levels of these bacteria almost certainly represents an STD. Now, what about other anatomic sites as reservoirs? I'm going to talk about that. And then I just also want you to forget that other things are often involved in sex. Semen has an alkaline pH that can tip some women over the edge in more ways than one. If you think of that graph that I showed you before where those women were marginally compensated with very marginal levels of lactobacilli, high levels of anaerobes, you give an exposure of pH that's about 7.5, you could conceivably maybe tip them the other way. And vaginal lubricants, both Rebecca Brotman's group and ours, have shown a relationship with BV and lubricant use. Lubricants not only have a lot of antimicrobial action, but the pH is often around 7 or 8, so very, very potent pH. The last thing I just want to point out is what I think is a fascinating analysis of the extravaginal reservoirs of these bacteria, because again it gets to this idea that when we are intimate with someone, whether it's through having sex or sharing anything that can relate to our microbial environment, we may get more than we bargained for. So what we looked at was colonization of extravaginal sites, namely the oral cavity and the rectum, as a predictor of subsequent BV in women who didn't have BV. So in other words, if you were colonized with these bacteria at these sites, did that mean anything? Was it a marker of recent sexual acquisition, or was it something else? So we did a case control study, as shown here, just published in JID. There aren't a lot of women, because you could imagine how hard it is to identify women who are BV negative, who you can follow over the course of a year, which we did, for incident BV, in whom you can do very complicated intensive taxon-directed PCR applied to oral, vaginal, and rectal swabs. We had already developed, or David had developed, the vaginal swab, but the oral cavity is a completely different animal entirely, huge numbers of bacteria in the mouth. So it was a little bit very, very, very challenging for his lab. What did we find? Sorry that these tables are a little bit small, but I'll show you basically what this means. These are data for what we found in the mouth. When you apply Gardnerella vaginalis in PCR form to the mouth of women who subsequently developed BV, at their baseline visit when they were BV negative, they all had Gardnerella in the mouth, which we thought was quite interesting, 15 of the 15. Only 45% of them had Gardnerella in the mouth if they did not develop BV. Still, it's a lot of Gardnerella in the oral cavity. So it is there, it's hanging out, but it seems to predict BV in this subset of women. I think also fascinating, although not quite as significant, is that we found Lactobacillus crispatus in the mouth, and it was more present, more likely to be present in the women who did not develop BV. So kind of interesting. There was a direct association with BV development, not only when you looked at it qualitatively, but also when you looked at it quantitatively. So when you look at the qPCR over here, these are the levels of Gardnerella in the mouth of the cases, and these are the levels in the controls. And you can see them. Some of the controls had a good number, but a majority of the controls had absolutely no Gardnerella vaginalis detected. So I think that's really interesting. When you look at the anus, essentially you see a very similar pattern, although you find more bugs. 15 or 100% of the people who went on to develop BV had Gardnerella vaginalis in the anus. So perhaps that's not as surprising to you. It's just maybe a marker. But remember, these women didn't have BV when this was detected. There had to be some transfer or inoculation, would be my guess. And Lactobacillus crispatus, as has been shown by others, us and Sharon Hillier's group previously, was protective, if you want to put it that way. That's a little bit too causal. But it was associated with a reduced risk of getting BV. And then you saw a couple of other bacteria that I mentioned before, Megaspira, BV82, Lepto, and Smithia, as being directly associated. So what does this mean? It could mean a couple of things. We think it could be that people were concomitantly inoculated sexually by these bacteria at multiple sites. So they got it in the anus, either through vaginal sex or anal sex. They got it in the mouth through oral sex, who knows? Or it could mean that these are people who cannot control these bacteria at mucosal sites. So you can't control it in the vagina, you can't control it in the mouth, you can't control it in the rectum, because that's your host immunity. We really have absolutely no idea. The obvious question is, can you get the partners of these folks in, and can you relate detection of these bacteria to exposure and acquisition and then incident BV? And those are the studies that we're starting to do now. So very, very interesting. So I would say that evidence that BV is sexually transmitted is insufficiently conclusive, but the weight of the evidence is supportive, and it really depends. Depends on what you're doing and who you're doing it with. I do think that the vaginal microbiome may in part be defined by the types of sexual exposure to partners, genital or oral communities. I didn't show you this, but we have very interesting data, and Kat Broadshaw's group just has published recently in PLOS ONE data to say that the microbiome of women who have sex exclusively with women is probably different than those who have sex with men. And I can come back and talk about that some other time. I think that dysbiosis basically, and dysbiosis again, is defined as a disruption of what's an optimal microbiologic milieu for use. May involve an exchange, then maintenance of bacterial communities. And the last data slide I want to show is something that Laura Sakuro and David's group is presenting right now, I think, at the American Society of Microbiology. I'm not sure if it's over. It may have been last weekend. And this is where I think it really gets interesting when you're thinking about the etiology of syndrome that we are concerned about with STDs. So urethritis, cervicitis, endometritis, pelvic inflammatory disease. These are too small for you to read, but you'll just have to trust me that BVAB2, which is now called Megibacillus indolatus, it's been cultured by Sharon Hillier's lab, actually has been consistently associated and significantly associated with both with actual clinical cervicitis, as visualized by mucoperial and cervical discharge, or very high numbers of polys on gram stain. And that has held up even when you control for some of the other bacteria in the mixture. It also doesn't matter whether you look for it in the cervical swab or the vaginal swab, which is shown here. So what this says is that there may be communities that are associated with pro-inflammatory potential that could be presenting as some of these syndromes. And what does that say for our approach to syndromic management, which we always don't think works very well? We also know that when you use very specific pathogen-directed PCR, such as Charlotte Gatus has done and others, to syndromes, you still end up with almost half of the pathogens, quote-unquote, unidentified. So if you apply all the known PCRs for all the known pathogens to somebody with cervicitis, or a bunch of them with cervicitis in our clinic, you will not find an etiology enough to have. And that's where I think we have to shift our thinking a little bit and think about what really is causing some of these issues that we're seeing. So moving forward, there was really a fun, I mean, I would say this was fun, but I thought it was really fun. We had a great workshop that Carolyn Deal and Joanne Schulte at NIH sponsored at the end of 2008, and we summarized that in the STD paper for those of you who really want to know the details, where we talked about the research gaps, and I think you probably have intimated what a lot of them are just from this talk. We need to use consistent definitions. I think we need to look at symptoms carefully, really try to understand whether women with symptoms are different than those without symptoms. We need to have consistent definitions for what cure is and timelines, and that's all laid out in this paper. I don't think we're going to be able to get around the complexities of time. So we do need to do frequent sampling. This is a very dynamic, very responsive system, like our bodies generally are. I mean, think about the gut. And I think we need to do that with inclusion of sensitive and specific behavioral data. And this is where the metadata idea comes in. You'll hear people talk about this term metadata. Metadata is that scary slide that I showed you, but then think about good behavioral measurements about what people are doing on top of that microbiome measurement. So not only what you did in terms of sex, but maybe what you did in terms of diet. You know, because clearly what you eat probably affects, obviously it affects what's in your rectum. Does that also affect what's in your vagina? It could easily do that. So that's really the idea of integrating studies of behavior and microbiology and then thinking about the role of host immunity. So I think it's a perfect paradigm for intensive cross-disciplinary approach. People really need to be open. I think they need to think about bringing the best methods of measuring each of these parameters to this. I can tell you, having sat in the room with some molecular biologists who are trying to study the behaviors that are involved here, they need us. And we need them very badly. And I don't mean that in any kind of pejorative way. It's just that we all come from very different perspectives, and it's a really great example, I think, of what we need to do. New product development, I think clearly we need better treatment modalities. That's a whole separate talk. So I think there are some answers, but we're left with the fact that BV is a syndrome associated with the acquisition of complex vaginal communities, includes many uncultivated species. I think that the heterogeneity of those communities could explain some of the differences that I talked about, but we don't know, and we need to think about some of the other factors. We can't neglect to study the host. This association with HIV transmission as well as acquisition, I think, is fascinating. Could you look at periodic presumptive therapy or some maintenance of normal vaginal flora to modulate that risk? Studies are underway. And I think I'll stop there and take questions, and again acknowledge not only David's team, but also Jared and Craig, who provided the slides summarizing the partners and interventions. Thank you. Minutes for questions, I think. Dusty, would you let me just take questions? Okay. Does anybody on the phone have questions?
It is *1 to ask a question,
okay.
And 1 moment.
Oh, we're waiting. Is anybody in the room? I'll interrupt the person in the room when you come on. But anybody in the room have a question? Comment? Stunned you all with those scary slides. Trust me, they are scary. I won't say who, but somebody told me this morning that they thought this was an arc. Promised that I would try to persuade them that it wasn't arcane. It's complicated. I think they meant it as a compliment. Any luck getting somebody on the phone?
At this time, there are no questions.
Oh, sad. Very sad. Anybody else in the audience? Anybody think it's interesting and crazy? I think the HIV data are really going to turn out. Well, I can be here for another minute or so, and then I must run to the airport. But other than that, thank you very much for your attention.
This does conclude today's conference. Thank you for attending. You may disconnect at this time.